Hello everyone, my name is Yasser Mohseni and I'm in the third year of my PhD at the Laboratory of Computational and Quantitative Biology at Sorbonne University. My supervisors are Professor Alessandra Carbone and Professor Elodie Lane. Today I want to talk about our novel computational approach called Deep Local Analysis, which evaluates protein docking conformations using geometric deep learning. So as you know, protein-protein interactions are essential for cellular machinery, and it's very important to understand how proteins interact with each other. As you can see in the left figure, a protein might have multiple interfaces, and using each interface, it can interact with different partners. So in the right figure, we can see a protein. Using one of its interfaces, it interacts with multiple partners but at different levels. For example, with this partner, it interacts with a high binding affinity, and with other partners, it has lower binding affinity. If we have a set of proteins, we are interested that who interacts with whom and in what manner. To answer this question, we use complete cross-docking approach in which we perform a docking algorithm on each pair of proteins from the set of proteins. And this results into a matrix that you can see in the right. So each cell of this matrix belongs to a pair of proteins, and it contains different ways that these two proteins can interact with each other. We call a way of interaction a conformation, and it's very important to study and evaluate all these conformations to find those that are near native conformations. This analysis will help us for the partner discrimination, and we used complete cross-docking experiments for both partner discrimination and the identification of the interaction site. And we published our results recently. So back to our original question, if we have two proteins, how they can interact with each other? To answer this question, the docking and scoring paradigm explores the conformational space, meaning it explores different ways these two proteins can interact with each other. And then it scores and ranks them based on the physical, chemical, and geometrical properties of the proteins. So in summary, protein-protein docking tries to predict protein complex structure from the structure of the monomers. And there is a competition called Capri in which different teams try to predict protein complex structure. So in this project, we want to develop a classifier to distinguish between near native conformations and incorrect ones. So how do we assess a quality of a conformation and classify it as a near native one or incorrect one? We do this using some quality measure and criteria defined by Capri competition. Here I will show you using an example. So imagine that we have a pair of protein. At the left we have the ground truth structure, which is basically the experimentally resolved structure, and at the right we have a conformation for this pair. We define three quality measurements. The first one is the root mean square deviation of the ligand. Next, root mean square deviation for the interface. And the last one is the fraction of native contacts. And there are some criteria from Capri that can classify a conformation into the incorrect, acceptable, medium quality, or high quality using these quality measurements. Another quality measure that got a lot of attention recently is called DocQ. DocQ combines other quality measures and produces one score for a conformation, which is a continuous score. It's between 0 and 1, and using this score, it classify a conformation again into the incorrect, acceptable, medium, or high quality. So to train our classifier, we use 2 complete cross-docking experiments and results conducted and provided by the lab, which includes 400 protein chains and 900 protein pairs with experimentally resolved references. These pairs, they have over 27 million conformations. In order to process and assess all these conformations, we combine 2 toolkits. One of them is called Int Builder, developed by the lab, and the other one is called Rapid RMSD. And after evaluating all these conformations, we create a train set including over 300 protein pairs, in which we could find about 4,000 acceptable conformations, and we kept 6,000 incorrect conformations. We also separated 20 protein pairs as the blind test set. In order to train our classifier, first we need to represent the structure of the conformation. There are various ways to represent a protein complex. This can include a representation of the graphs and then using graph attention mechanisms, or a representation of the protein complex as a point cloud and then using SE3 transformers. But one of the representations can be volumetric map, in which we put a grid on the interaction site to capture the arrangement of atoms in the interface, and then we convert this arrangement into a density function, as you can see in the middle. But this representation has a problem. It is sensitive to the rotations of the conformation, so it's not rotation invariant. To resolve this problem, we adapted an approach called ornate, in which an amino acid and its environment are represented with the locally oriented cubes. These cubes are centered and oriented using the common scaffold of the backbone of the amino acids. And thanks to this local orientation, this representation is rotation and translation invariant. Also, thanks to the local representation, we will have enough training data which is necessary for the data-hungry deep learning approaches. Another aspect of our methodology is the classification of the interaction site to different regions based on the changes of the solvent accessibility upon binding. We can classify the interface into 3 main regions. They are called support, core, and rim. Support includes the residues that they are largely buried in the monomers, and they become more buried upon the formation of the complex. The core region includes amino acids that have solvent accessibility in the monomers, and they become buried upon binding. And rim represent the amino acids that they have solvent accessibility before and after the formation of the protein complex. These motivations have led to the development of DLA ranker, which stands for Deep Local Analysis Ranker, in which we represent the interface of a complex as a set of locally oriented cubes. Each cube contains the local arrangement of the atoms in the space, which eventually be converted to the density function here. And we give this density function, this volumetric map, to a series of the conventional 3D convolutional kernels. And then the result is a score representing whether the residue, the central residue, is comfortable with respect to its environment or not. Also, as I mentioned in the previous slide, we use the regional classification of the interface as auxiliary features for our purpose, for the training and test phases. Since we are treating each interfacial residue, each of these cubes independently, we need to find some aggregation schemes to aggregate the scores coming from different cubes for the different interfacial residues and put one scores on the whole interface. That's why we use the graph-based aggregation and average and other aggregation scheme called averaging schemes. So I won't talk about the graph-based aggregation in this presentation. I will focus on the averaging schemes in which we average the scores for either all the interfacial residues or those belonging to the core and rim regions or those belonging to the support and core regions. So as I mentioned before, we had put aside 20 protein pairs for our blind test set. Each of the conformation for these 20 protein pairs are generated using the coarse-grained rigid body docking algorithm called MaxDo. And we chose the best 1000 conformations, decoys, chosen by MaxDo, and we will try to re-rank them using DLA ranker. The blue represent the result of DLA ranker and pink represent the result of MaxDo. So in this slide, I will compare the performance of DLA ranker and MaxDo in finding at least one near native conformation at top rank positions. Below we can see the 20 protein pairs colored based on their level of difficulty. We determined the level of difficulty using the deformation and deviation between the pairs and the experimentally resolved reference. So as we can see, for the majority of the protein pairs, DLA ranker can find the near native conformation at the top first position, and for others it can find at least one near native conformation at the top 5 or top 10 positions, and clearly it outperforms MaxDo. So here I will talk about the impact of using different averaging schemes. At left we can see a plot in which we compare the performance of DLA ranker when we use different averaging schemes. So we have the performance when we used only the residues from core and rim, only the residues from support and core, or when we do averaging over all the interfacial residues. And at the y-axis we have number of pairs among 20 protein pairs for which DLA ranker could find at least one near native conformation at various top rank positions. And we can clearly see that when we apply averaging schemes over the interfacial residues from core and rim, DLA rankers outperforms all other averaging schemes. And at the right we have an example of how DLA ranker performs in comparison to the MaxDo. So we have a structure of the receptor and ligand. Below we have the structure of the receptor. The white one is the docking receptor and the black one is the experimental receptor. Above we have again the black one, which is the experimental ligand, and blue one is the best ligand pose found by the DLA ranker. And as we can see, we have a good superimposition with respect to the position of the ligand. However, the MaxDo find this ligand position as the best ligand position, which is already far from the experimental ligand position. We compared the performance of DLA ranker with two other deep learning approaches. The first approach is called deep rank, in which the interaction site is represented with a global rotation variant grid. Deep rank uses data augmentation to compensate the fact that the representation of the interaction site is not rotation invariant. The database of this approach is the 142 dimers of the fifth version of protein docking benchmark. DeepRank generated near 500,000 conformations with different docking, ab initio, and interface guided scenarios using Haddock algorithm. Following author's report, we performed tenfold cross-validation and reported the results in the enrichment factor. In the y-axis, we have the enrichment factor. The enrichment factor is the fraction of acceptable conformation found at top rank position for each target complexes. And here we have the median across all the target complexes. The solid line is the median, and the shadow lines is 25 to 75% percentile. And we can see that DLA ranker outperforms deep rank, particularly the averaging schemes of SCR and CR, the black one and the purple one. It is worth mentioning that DLA ranker's training process is more efficient than deep rank because it's rotation invariant. It doesn't need the data augmentation. Another approach is called DOV-GNN, in which the complex interface is represented by a graph, and it applies graph attention mechanism to process these representations. The performance of DOV-GNN is evaluated on the DocGround database, including 58 target complexes. We followed author's experimental setup and performed four-fold cross-validation. Here we have the performance on the first fold, which is reported in hit rate. Hit rate is a measure, is calculated as follows. It's a fraction of target complexes with at least one near native conformation at top rank positions. The averaging scheme of CR outperforms the performance of DOV-GNN. We also compared the performance of DLA ranker with the combination of Class Pro and AlphaFold 2, which is designed to improve docking of protein models. The performance is done on the same 142 dimers, and as we can see, DLA ranker with the averaging score of CR outperforms Class Pro AlphaFold 2. In terms of finding a number of target complexes with at least one near native conformation at top rank positions. Here we have 105 in total until top 10, and Class Pro AlphaFold 2 has 94. DLA ranker is also proved useful to detect alternative interfaces. In this experiment, we used a homodimer and we generated 3,000 conformations using attract docking algorithm. And after analyzing all these this pool of decoys, this pool of docking conformations, DLA ranker could detect three different alternative interfaces for this homodimer. I mean, three different ways that that this protein chain can interact with itself. So in this figure, the red regions represent the alternative interfaces. It includes the residues with the highest score according to the given by the DLA ranker. This is in comparison with the alternative interfaces found by the binding propensity, which also process the pool of conformations. In the binding propensity, we could find only one interface, and the other interfaces are neglected by the binding propensity. So DNA ranker is available on the GitLab of the laboratory, and it's accessible for everyone who is interested to study and evaluate the conformations of the protein complexes. Also, we are participating in the current edition of the CASP for the quality assessment of the predicted protein complex structures. At the end, I would like to thank my supervisors, Professor Alessandra Carbone and Professor Elodie Lane, for their support and supervision. Also, our colleague Simon Causet for his help. Thank you very much.